Welcome, everyone. Thank you for joining us for today's webinar, USP Particulate Testing. My name is Amelia McKinley, Multimedia Specialist at Entegris. Entegris is a leading provider of products and systems that purify, protect, and transport critical materials used in the life sciences and semiconductor device fabrication process. Just a little housekeeping before we get started. If you have any questions during the presentation, please type them into the question box in your control panel. Time permitting, we will answer them during the Q&A session at the end of the presentation. If we happen to run out of time before answering your question, we will reach out to you via email after the event. Also, please note we will be sending out a recording of today's webinar to all registrants. And now I'd like to introduce today's speaker, Mark Bumiller, Technology Manager. Mark has worked in the field of particle size analysis for over 35 years. He has held a wide variety of positions, including Product Manager at Hayek Royco, Technical Support Manager and Vice President of Business Development at Malvern Instruments, VP of Particle Products at Horiba, and Technology Manager at Particle Sizing Systems LLC, and now Entegris. He has served as a member of the Expert Committee for USP 788, the Executive Committee of the International Fine Particle Research Institute, and the Executive Committee of Particle Technology Forum of the American Institute of Chemical Engineers. Mark is an active member of Technical Committee 24 within ISO, helping to write standards for particle size and zeta potential analysis. So now, without further ado, I will turn the time over to Mark.
Thank you, Amelia. And today we're going to discuss something that I've been involved with for a long time and I find fascinating, which is how we do particulate testing per certain USP tests. All right, so here's a quick outline. First, we're going to talk about these three tests, 788, 787, and 789. With these tests, we're measuring contamination, particles we wish were not there. This will be in protein, this will be in parenterals, proteins, and ophthalmics. Then we'll talk about USP 729, where we're not looking at contamination, we're looking at the actual drug product, which is a lipid emulsion. When we first implement these systems into laboratories, a very important phase is the validation and verification. We'll cover in some detail how we go through that process. And then we'll talk about how we've come up with new solutions for automating these tests for labs with a lot of sample throughput who would like to see how to perform these automatically. So how do we define a subvisible particle? Well, there's the visible range and there's the subvisible range and this gray zone in between. And where this difference is depends basically on your eyesight and how old you are. I know personally I can't see down below 100 microns. Some people might be able to. But in general, we talk about subvisible particles being something down below about 100 microns. The size ranges of interest for most of these tests are at 10 and 25 microns, very solidly in the subvisible region. And I just pointed down here the range of the sensors we'll discuss today. For instance, the LE400 sensor measures from 0.5 to 400. Across a broad range from the subvisible into the visible. And then we have the FX nano sensor if we need to measure down to 150 nanometers. On the table on the left, what we're showing is some of the known sources of contaminations that show up in these products, some of which are expected, such as maybe some glass fragments or some metal particles. A lot of products have silicone oil droplets in them, to the surprising ones, such as insect parts. So this is just a range of where these contaminants come from, adapted from a book written by Tom Barber, one of the original experts in this particular field. Here are the basics of what goes on in the USP 788 test. The real focus is at 10 and 25 microns, and the number of particles per ml, or per container, in these size ranges. How we perform the path-fail calculation depends on the size of the vial that the drug is contained in. We'll define a small volume parenteral vial as having a volume under 100 ml. And here, since we have smaller volumes, what we'll do is we'll combine multiple vials, and then we'll measure that combined volume. We will measure it 4 times, delete the first, and look at the average of the last 3, and then do this calculation to determine the number of particles per container, greater than 10 and greater than 25 microns. A large volume parenteral is defined as a vial where we have a volume greater than 100 ml. And here we might be able to actually sample directly from the vial. So here we're going to base the pass-fail criteria on number of particles per ml, greater than 10 and greater than 25, using the values you see here. Here is the instrument we're going to use for the data we'll show you today. This is the AccuSizer single particle optical sizing system. There's a sampler for moving the sample from the container up through the sensor. There's the sensor where all the action occurs. This is where we take the measurement. And there's the counter, which is a pulse height analyzer. On the graph on the right, we see a diagram of the sensor. This particular sensor has two detectors. It has an extinction detector. And when we're using the extinction detector, we call this a light obscuration test because we're looking at the amount of light blocked by each particle as it passes through the measurement zone. And that detector can detect particles down to about 2 microns. If we'd like to measure smaller particles, we've added a second detector, a scattering detector. And this allows us to measure down to 0.5 microns. We'll explain later on some of the advantages of being able to measure down to these smaller sizes. We take the signal from these two detectors, sum them together, feed them into the counter, and then through the use of a calibration curve, we convert each pulse into, we count and size each particle and build up the result. So this is the AccuSizer SIS system, which we typically use for making these tests. Here we have a video of watching the AccuSizer SIS in action. As we watch the video on the right, what we'll see is as the syringe pulls down, it's going to aspirate the sample through the sensor where the measurement is made. After we've completed that measurement, we'll then push that sample either back out to waste, or we could conserve the sample and push the sample back into the original beaker. This sampler has no dilution, so we use this typically for contamination and for low concentration samples. And the volume accuracy here is very solid, all the way down to being able to measure volumes as small as 150 microliters, which we will discuss when we talk about USP 787. Here we're saying the protocol that we use in the software to set up a measurement. We'll choose a sample volume, and typically for USP 788, that will be 5 ml. We'll choose a number of replicates, that typically would be 4. A tare volume is how much do we pull before we begin the measurement. With some samples, we want to be very careful to separate any diluent from the actual sample, and we can introduce an air volume in between those two liquid volumes to avoid any cross-contamination. There's a button here to work for very low measurement volumes. We essentially change how we're going to interact with the syringe. We can either push the sample to waste or preserve the sample, and if we're working with very low sample volumes, that might be very advantageous. Also, if we're working with very low sample volumes, see here, push-pull mixing. This is a new innovation only in this system, where instead of using a stir bar or manual mixing, what we can do is pull the sample up with the syringe and push it back several times. That gives us a very good mixing of the sample without requiring any other mixer involved. If we want to just work down to about 2 microns, we could work in extinction mode to get down to 0.5 microns. That's when we use the two detectors. That is summation mode. And we can have the system automatically flush down to a predetermined concentration, either based on the concentration or on time of flushing. So this is the first tab of the protocols that we use to set up for the measurements. Here we're looking at the second tab where if we're performing USP 788 for small volume parenterals, we would enter the volume of each container and the number of containers. And if we enter that information, then when we get over to the report tab, then we would have the ability to run the reports for the small volume parenteral testing. Up above, we see where we would enter the information if we're doing USP 729 testing to enter the information on the lipid emulsion. The third tab in the protocol is where we can choose to set up automatic reporting. For instance, if we're doing standard USP 788 testing, what we can do is ignore that first measurement and then take the average of the next 3. Then we can automatically create the report you see down here on the right, where we look at the number of particles either per container or per ml at 10 and 25 microns for the second, third, and fourth run, look at the average values, and then get an automatic pass-fail for each sample. So those are the 3 tabs in the protocol that we can use to automate these measurements. Now, on top of USP 788, there's another chapter in the USP 1788, which gives us a lot of information in terms of how can we set up the system, how do we verify the system, how do we get ready to make measurements, including tests such as, let's make sure if we're going to ask to pull 5 ml, it is really 5 ml. So there is a sample volume accuracy test, which must be within 5%. We calculate the sample flow rate. We need to test the calibration curve, which you see here. This is the curve in the table for the calibration. We need to test that on a regular basis, either every 6 or 12 months. There's a sensor resolution test and a count accuracy test. This is a calibration kit we use for calibrating the systems. And according to USP 1788, we must check the calibration curve at least 3 points. When I visit customers and do these calibrations, I'll typically check 9 or 13 points. We do everything a little more than what is actually required, but at least look at 10, 15, and 25 microns. The 10 and 25 microns are because those are the basis of the USP test. I will typically also measure at 15 microns because the USP count standard, which we'll need to analyze to verify the system, is actually a 15 micron polystyrene latex standard. And what we need to do, if you look at this graph on the right, since it's a 15 micron standard, the count accuracy test looks at the counts greater than 10 and greater than 15 microns. So we need to have these points on the calibration curve very accurate to be able to pass the count accuracy test. And actually, the accuracy at 15 microns really defines the pass-fail criteria, because there's also a calculation of the counts at 10 and at 15 microns. And if this peak isn't clearly defined, you might not pass that ratio test. The sensor resolution test is basically saying, are you working with a good sensor? So when we look at the standards we use, no standard is perfect. There's a particular spread to the standard itself. So we call that the standard deviation of the standard, which comes from the certificate of analysis on the bottle of the standard you're using. Then when we do a measurement, no sensor is perfect. So you somewhat broaden that distribution due to imperfections in the sensor. And there's a calculation you see here where we need to have this value below 10%. All we do is when we measure the 10 micron standard to perform the calibration check, we now have a report we can run where it will automatically calculate the sensor resolution test. In my humble opinion, I don't know why this is still in here. Back in the 80s, when I was doing this at another company, we were actually using white light sensors, and some of these failed. But as soon as the industry switched over to using laser diode sensors, certainly I've never seen a sensor created by Entegris that has not passed this. But I recently wrote an email to USP to explain this to them, and they told me, yeah, go away. So I don't see this test disappearing anytime soon. Here's a picture of the USP count standard that you can purchase directly from the USP or from some other sources, such as Micromeasurement Labs or from Thermo Fisher. I used to be worried when I ran this in the field because each of these bottles costs about $650. And partly because of this anxiety of people in the field, we figured out a way where you can run it once and then make sure that that you pass by making appropriate adjustments to the calibration curve. So this is a sample where we need to make sure that we pass at the number of counts greater than 10 and the ratio between 10 and 15. We make this measurement now, and we can have this automatic report print out at the end to show us that we passed the requirements based on the certificate of analysis for the standard. When we first install the systems, one of the first things we do is sort out the user management, because we want people who use the system to have their own password. Then we might also want different users to have different levels of authorization. So we'll set up a new user, give them a name, decide what they are authorized to do. Can they run the instrument? Can they sign and approve results? Can they change protocols, etc.? Are they allowed to sign reports, is what is set up here. And then we can also organize the basic security settings. So these are important functions that we'll sort out before we start having the operator using the system. At the end of each measurement, we can have someone review the report and sign the report or reject the report. And these are the features where we can organize who is authorized to sign and who can approve or disapprove results. In the last two slides, we've described some of the implementations of 21 CFR Part 11. This is the official document that tells us how to organize our compliance with keeping track of electronic signatures, electronic records, and copies of results. Now, we have created this document to help customers understand our implementation of 21 CFR Part 11. We go through each of the requirements, and then we explain how we've implemented each of the features required in the official 21 CFR Part 11 document. This helps customers understand our approach and also usually helps their internal validation. Another document we have created to help the internal validation of the system is our description of our approach to software testing and verification. This is the document you see on the left. This is a fairly in-depth and long document. You can see it's 282 pages long. It goes through how we fix errors and also how we take our approach to validation. And for every calculation we make in the software, we compare a manual calculation in Excel to the automated reports. We do this multiple times, and we have verified every calculation we make in any of the reports you see in our system. This typically greatly facilitates the internal validation of the system and gets the system up and running quicker at the customer site. Now let's talk about USP 787, which is very similar to USP 788, but is specific to when the drug is a protein as opposed to, for instance, a small molecule. And some of the differences here are some of these protein drugs are really running up the concentration, like up past 300 mg per mil. And since we get up to those very high concentrations, the protein itself might actually be at such a high concentration, we need to dilute the sample before the measurement. Since we're talking about dilution, there's some other comments about sample preparation. And sometimes the volumes of these protein therapeutics are so small, we need to get away from requiring a 5 ml measurement. And we may be doing measurements just on like 200 microliters. In terms of mixing and doing the testing at these small volumes, that really can become more of a challenge. And so we've had to optimize some settings in the system to make sure we can still perform very accurate and repeatable tests at these small volumes. This is data from a study we performed to really determine the smallest sample volume we're comfortable measuring. We took the USP count standard, which has a known concentration, and we reduced the sample volume from 650 microliters down to 50 microliters. We looked at the counts in each measurement, calculated back to the counts per ml, and checked the accuracy here and the repeatability, defined as a coefficient of variation or the percent RSD. The plot on the right shows you we have a very linear response, and we actually could perform very accurate measurements down to 50 microliters, but we don't typically recommend that to customers because there the mixing and some other challenges get fairly difficult. So we usually say, measurements down to 150 microliters, very comfortable, and we can prove we have accuracy and repeatability down to that low of a volume. The last contamination test we'll talk about is USP 789. Here we're talking about ophthalmic solutions. Very similar to the other tests, we're looking at contamination greater than 10 and greater than 25 microns. Here we just look at the number of particles per ml, and you see the specifications here of 50 per ml at 10 and 5 per ml at 25 microns. And we have automated tests for USP 789 and the software as well. Many of these measurements are still performed manually in the lab, but for laboratories where there's a requirement for putting a lot of samples through the lab every day, we have two solutions for automation. On the left, we see the microplate analyzer, which is what we would recommend for doing USP 787 testing of therapeutic proteins. And on the right, we see the autosampler, which is used for standard USP 788 testing. Here we'll see the video for the microplate analyzer. And what happens is the probe will go down into a washing station and then move over to one of the wells in the microplates you see over here. We'll sample from that microplate, aspirate the sample through the sensor for the measurement. Once we've completed that measurement, the system will move back over to the cleaning wells, clean up, then move over and do the measurement on the next well in the microplate. This is a solution for low volume testing. Here on the right, we see the auto sampler for USP 788, where we have larger volumes. And here is very similar. A probe comes over, aspirates the sample up through the sensor for the measurement. After we've completed the measurement, it'll move over and clean the probe, then come back over and measure each of the other tubes or bottles set up in the sample rack. These solutions for automation are actually making life a lot easier for customers who have many, many samples to analyze, and these are rather unique features to Entegris. I recently used the microplate analyzer to perform some tests on a protein, a monoclonal antibody. Here we're looking at the protocol that I used, where actually I had the measurement volume a lot lower than what you see in the protocol. I was doing 4 replicates out of a very small well, and I was measuring 250 microliters 4 times. And the only way I could do that is using the sample preservation option that you see here and push-pull mixing. So what the system does is it pulls some of the sample up and pushes it back, performs the mixing. Then after I pull up the 250 microliters for the measurement, it returns it to the same well, and then we'll do another measurement. These are the settings you can set up for the protocol. We could enter the number of particles per container if we wanted to do small volume USP 788 calculations and automatically do the reporting as you see here. The sample I was measuring here is actually this NIST standard, which is a monoclonal antibody based on IgG. And this is about the only NIST standard I know out there if you want to work with a protein that's really stabilized and you know it's very clean. A lot of the other ones I see, there's a lot of contamination. And when you receive this standard from NIST, it comes frozen, and then you thaw it, and then you perform your measurements. And the first thing I did when I received this new standard is I compared the new standard to one that I'd bought about 2 years ago. And after I had thawed it, I froze it again. So the results you see on the right is looking at the new standard, which is in blue, which I just thawed that day, and then the older standard, which you see in red, which is one that is from 2 years ago. And sure enough, we see this increase in particle count across these size ranges, and that's indicating there probably has been some agglomeration that's occurred because of the freeze-thaw cycle. Now you notice in the table here, at 10 and 25 microns, there are no particles whatsoever. This is a very clean protein sample. So both of these would pass USP 787, and sure, 10 and 25 microns is where the test is written around. But if you really want to see the difference between the degree of aggregation, you really want to look at smaller size ranges. And the really interesting data comes here underneath below 1 micron. And the unique feature of the LE400 sensor is this ability to do this test up at 10 and 25 microns, but also provide data down to 0.5 microns. And if you're working with proteins, this system can also be used to look at degree of aggregation, which is very important, down at these smaller size ranges. Here I'm looking at the repeatability for the 4 measurements. We're going to discard the first and then look at the average of the next 3. So this is showing how repeatable these measurements are, even using this very small sample volume. Here we're doing the overlay plotted in the AccuSizer software. Then I exported the data to Excel and I put in my error bars showing the repeatability of these measurements. And down here at the smaller size ranges, you see the coefficient of variations are extremely small, showing excellent repeatability. Up at the larger size ranges, the number goes up. That's just the statistics of small numbers. So this is showing the ability to measure down at smaller size ranges very repeatably when working with protein samples. The last test we'll discuss is USP 729 for lipid emulsions. And here, remember, we're not looking for contaminants. Here we're looking at the actual drug product, which is a lipid emulsion. This test has method 1 and method 2. Now, these lipid emulsions have a mean size down near 0.35 microns, 350 nanometers. And method 1 is to use laser diffraction, or we typically use dynamic light scattering to look at the mean size. Then method 2 is looking at the tails, the large particles up here greater than 5 microns. And there's no instrument that can do both test 1 and test 2. Here we're going to use dynamic light scattering for method 1, and then the Accusizer system for method 2 to determine up here these large droplets, because these large droplets are both a health hazard, and also the large droplets are a very good indication of instability. The images you see down here on the left is showing a good versus a bad. This is an SEM photograph where we have very few particles greater than up around 5 microns. And then when we see more of these, then we know this emulsion is destabilizing. So method 1, we're going to use dynamic light scattering. Dynamic light scattering is based on the Brownian motion. The video you see on the right shows the red particle being bombarded by the blue water molecules. And that random thermal motion can actually generate some light scattering. Small particles move a lot faster than large particles. And what we'll do with dynamic light scattering is we're going to look at the scattered light due to that Brownian motion. We have a light source, we have a detector, and this detector is just counting photons from the scattering of the Brownian motion of those particles. We feed the rate of those photon counts into a function called an autocorrelator. From the autocorrelator, we can determine the translational diffusion coefficient and then use this equation to calculate the mean size. And this is the NICOMP DLS system, which is used around the world for performing this test. Here is a result for method 1 using dynamic light scattering. Here we can see the mean size was 313 nanometers when actually it just needs to be below 500 nanometers. This is a result from DLS for method 1. The requirements in this test, typically it says measure at 90 degrees, do a verification at several sizes, do a dilution because these are always very concentrated samples, and the NICOMP has a unique feature of auto dilution to make that very simple. And then just look at this intensity mean, and that must be below 0.5 microns. And look at your chi-squared, and that should be a low value. So here's a typical result for method 1, looking at a lipid emulsion. For method 2, because these are very concentrated samples, we're going to use the Accusizer, but here we need to do dilution. What we see on the right is a fluidics diagram of a 2-stage dilution process we use. We fill a sample loop. That loop is injected into a dilution chamber. That's our first stage of dilution. And then as we draw the sample down through the sensor, we continuously bring in this filtered diluent line. They mix here and we perform the measurement. And we need to do this kind of dilution to fairly high dilution ratios. And this system can automatically create accurate dilution ratios you can never do with your hand. For instance, doing 1,000,000 to 1 dilutions very accurately. So this is the Accusizer APS that we typically use for method 2 in USP 729. Here is the Accusizer APS. And on the right, we see a video where we're drawing the sample from the beaker. We just colored this red so you could see it. We fill the sample loop, and then the loop is injected into the dilution chamber, and then diluted in a second stage of dilution before we pass through the sensor. All this is automated, making it very easy to perform USP 729 method 2 to calculate the number of particles greater than 5 microns, or what we call the PFAT5 calculation. Here's how we set up the protocols and automatic reporting. If we're going to do the automatic PFAT5 calculation, we need to enter the fat concentration in a weight volume, and we need to enter the oil density to get these calculations accurately in order to accurately perform the calculations. Then on the third tab in the protocol, we choose USP 729 reporting, and we can automatically generate the reports on completion of the measurement. So that is a summary of the newest instrumentation and automation available from Entegris for performing USP subvisible particulate testing. Some of the ways in which these systems are unique is first, with the Accusizer using the LE400 sensor, we have this wide dynamic range, measuring from 0.5 to 400 microns, and we can actually get down to 0.15 microns if we need to for testing protein aggregation. We only need to measure at 2 size ranges, 10 and 25 microns, right? But we actually have a counter with 1,024 channels. And you don't need that for the reporting, but when you do things like the sensor resolution test or honing in your calibration curve to pass the USP count accuracy test, that's a terrific advantage. We showed, as we talked about the automatic reporting and the calibration routines, the software we use to run the systems are the most advanced, making these measurements much easier and quicker for people performing these tests. And for people who need high throughput in the laboratories, we have two solutions for automation. We have an auto sampler for performing typical USP 788 tests, and the A2000 microplate analyzer for doing low volume tests when working with proteins following the requirements of USP 787. Thank you. That is a material that I created for today's discussion. Hopefully this was informative and perhaps brought up some other questions people would like to ask now. Now we actually have some time to have some interaction, and we'll have two possible interactions. Right now, Amelia is going to show on your screen some polling questions. And if you'd please respond to those, that might help get some discussion going. And while you're doing that, think of any other questions you'd like to ask. We have some time now for some Q&A to see if anything that I discussed today has brought up any questions or other things you'd like to discuss.
Thank you so much, Mark. Great job. Well done. And yes, we do actually have a few poll questions for our audience before we open it up to Q&A for Mark. So the first question is, which USP test are you mostly interested in? So you can select USP 788, 787, 789, 729, or other. And we'll just give you a few moments to respond to that before we close the poll and review the results.
Yeah, in terms of the customers I deal with, it's pretty heavily weighted USP 788. 787 is really getting more popular because a lot more of the new drugs coming onto the market are protein-based. And those measurements really are a touch trickier. Part of it is the low sample volume, but we've spent a lot of time investigating that. And actually, I'd submitted some data for an article that we could share if people want to see it, where it was really kind of focusing on what about this small volume testing. I even looked at the difference of the ID of the two bringing the sample up through the sensor to see if that shear might cause any problem with the particles. And the basic take home is not a whole lot, but you really have to be very careful when working with these smaller volumes. And we've kind of automated the system for people who need to get down into those low sample volume ranges. USP. Sorry, go ahead.
Oh, it looks like your typical results are proven yet again here. So it looks like, yeah, 788 is the highest.
Yep. So, this has been around forever. I only missed writing that standard by, I think, like 8 months. I joined the committee that was organizing that 8 months after it finally been written. And I wish I could say it's changed much since then. It really hasn't. I'd mentioned that sensor resolution test should be pulled out, but that's fairly well understood. Kind of the real changes are, in terms of the person who's in the field doing these things, what we've done with the software really makes running that count standard a whole lot easier. And what we do with calculations, the automation, the calibration as well, the real changes have been made to make life easier on the user. The basic requirements haven't changed tremendously through the years with that one.
That makes sense. So let's move on to our second poll question. How do you currently perform subvisible testing? Do you use the AccuSizer, another brand instrument, lab services, or none? And of course, we're hoping to see a lot of AccuSizer answers, but perhaps we'll see some others as well.
Yeah, the thing in technology first to market always makes a big difference. And back in 80, boy, I joined Hayek in 85. It was actually funny. Back in 84, they wouldn't do that now. There was actually an asterisk in USP 788 that said a system could be purchased by Hayek. They would never do that now. So not much has happened with that instrument. Actually 8 years ago, they're still selling the system I helped design back then. So not a whole lot has happened with that system, but there's still a lot of momentum just because of that history. We have by far the superior technology now, and we keep trying to increase our market share by providing a better solution to our customers.
Looks like the results are relatively evenly divided here with the other brand instrument leading. What's the advantage of using, your own instrument over lab services?
People usually use lab services when they're just getting started. They only have, 10 measurements a month or something like that. And then eventually the expense of the lab services grows high enough where they consider they want to bring the instrument in-house. And then there's the turnaround time. You send it to the outside service and you get the result back in 72 hours. Maybe you want to get it back now because I'm doing something with my process. So it's both monetary and it's a question of time. And people eventually always want to bring it in-house to do the tests themselves.
That makes sense too. All right, let's do the last poll question before we move on to Q&A. How many samples do you need to test per week? Less than 5, less than 10, less than 20, or more than 20?
Yeah, and I know customers who just use this for R&D and they use it 12 times a year. And I go to laboratories where they're going to do 24 measurements today. So I see a broad range with the number of samples people need to push through. And that's kind of, I also bring that up just to help people consider automation. If that number gets up over 20, 24 a day, then maybe the automation is going to start making sense.
All right. And there we go. So a lot of less than 20s in there. Less
Yeah,
than 5 is our biggest one though.
yeah, so that's not terribly unusual. But when I go to the lab, like the data I showed for that protein data, I don't live in Santa Barbara where we make these and do that work, but I go to Santa Barbara once a month and I generally will generate 400 results in about 3 days because I'm just data driven. And so when I go up there now and I'm going to try to crunch out 200 or 300 results in 3 or 4 days, I always use the automated mode. But I'm lucky I work for the company that makes it and they're right there.
Right. So let's move on to some Q&A. What questions do our audience members have for Mark? Remember, you can type those questions into the question box in your control panel. We have actually already received several excellent questions. So Mark, I'm just going to fire away here.
Go for it.
All right, here's the first one. Can the same instrument be used to make all 4 tests, the 788, 87, 89, and 29?
Yeah, boy, I wish the answer were yes. And I guess theoretically it's yes. The point is all except 729 are looking at contamination, which are low concentration. And you theoretically could do USP 729 using the SIS, the syringe sampler. But that would assume that you're such an amazing chemist, you can do 1,000,000 to 1 dilution accurately. And I challenge anyone to claim to be that person. I've tried it and I fail. So theoretically you could, but with 729, boy, are you gonna stretch your abilities to do those dilutions accurately on your own. And I would highly recommend considering the auto dilution features of the APS for that.
Hey, sometimes honesty is the way to go, right? You got to tell people how it is.
Oh, trust me, I'm flawed, including in the lab.
So the next question I have is, the new software looks very cool. Can I upgrade my existing AccuSizer to the new software?
Yeah, that depends. If it is an instrument in the last 8 years, certainly. If it's an older system, then it gets trickier. We never mention pricing in these public events, but it gets much more expensive to upgrade an older system is the way I'll have to leave that. And these instruments are bulletproof. I see instruments that are 15 years old all the time, and that would be difficult to upgrade is the way I'll leave it. And in Japan, when you hear it's very difficult, that means no. I'll leave it at that.
I like that. So the next one I have is, what is the minimum volume for making measurements using the microplate analyzer, and how many samples can it analyze in one tray?
Yeah, we talk about you theoretically can do 50 microliters, and I was doing 250 in the data I showed you. When I was doing those tests that I shared the repeatability data, I was making sure that my data was repeatable because I knew people were going to ask to see that. If you're going to drop that volume below 250 microliters, you're going to see your repeatability drop off. So I would say you can do good measurements to 150 microliters. You can generate great data at 250 microliters. And I think those trays are 96 wells per tray, and you can stack 2 trays on the system. But boy, do you have a lot of samples if you need 2 96 well trays. So yeah, I'll say 96 or so.
Yeah, so quite a few.
Oh yeah.
That's good. The next question I have is, how often do I need to recalibrate the instrument? Can I check the calibration myself?
I usually would say this is a risk-based assessment. If your entire plant runs on, does my sample pass today, I'd be checking that calibration daily. If I use the instrument for 10 samples a year, I'm going to have trouble convincing myself I need to calibrate it more than once a year. If you look at what the USP or FDA expect from you, some people say 6 months, some people say 12. I see 2 interpretations. So it's at least annually if you're going to say, I'm going to follow good laboratory practices. And you could test it yourself. And if you're one of my customers and you say, I want to test it, I'll show you how to do it. But most people like the paper trail of having the vendor come in and do it and put a sticker on there saying we tested it. So at least annually, and we'll show you how to do it if you want to do it on your own.
Excellent. Next question is, can you please elaborate on how particulate testing has been applied to single-use assemblies, like bioprocess bags integrated with tubing and connectors? Do integrators conduct routine particulate testing on their single-use assemblies?
I'll even broaden that bigger. So the answer is yes. And actually, the new application note I'm working on is just medical devices in general. And we could talk about single-use bags and things or assemblies as medical devices. So I actually have personally installed 2 or 3 systems this year for that application, both internally and Entegris. I mean, Entegris makes the Aramis bags, which are single-use bags, and they have been tested using what we used to call the particle sizing system, PSS systems, for years. So sure, that testing has been going on for years, and we do it internally at Entegris. And now I see a lot of customers using it as well to say, what is the state of cleanliness of a bag or of an assembly, of an IV set, et cetera. And all you really do is to make these tests is you do some kind of flushing, some way to try to knock particles off of the assembly, and then use the AccuSizer to count the number of particles per ml at 10 and 25 microns. And if you do it on a per ml basis, you could then bring that back to the pass-fail criteria in the large volume injection calculation in USP 788. So absolutely, it's being used for that. And that is a growing use. And probably within about 2 months, I'll have a new application note out on broadly how to do these measurements and how to automate doing them in the calculations. So yes, I have one application note on single-use bags on the Aramis we could share now. And give me a little time and I will have data for things like IV sets and other medical devices we can share. So yes and yes.
Wonderful. There was another more specific question about single-use assemblies, but I'm getting the signal to have that question be answered separately so that we can give you a fuller response, just so that that audience member knows. Now, the next question I have is, can you export data to Excel CSV files?
Yeah, so what I do, like I said, I go to the lab and I'll knock off 300 measurements in a couple of days. And the only way to organize that data is through spreadsheets. So
Mm-hmm.
when we're in the report tab, you can automate the report, such as USP 788 reports, but then you click somewhere else and you say, and also export it to Excel. and all that data automatically, each result turns into an Excel spreadsheet. And then I'll take those, group them together and organize them to do things like what I showed with the error bars, et cetera. So that's automated, and then it's up to you to take those spreadsheets and tie that together. But sure, that capability is there. And if you generate the volume of data I do, it's the only way to deal with it.
Wonderful. Next question is there automation software available for system suitability calculations for method 1 of USP 729?
Yeah, I haven't heard that question before. So if we're doing method 1 and we're using dynamic light scattering, and what we're going to do is say, can we automate that? And there's some things we can do other people can't do. And that includes, for instance, those, remember, are high concentration samples, and we can have an auto diluter built into the NICOMP. Secondly, we are the only company, Entegris is the only company that has an auto sampler that can work with the NICOMP. So we can do the automatic dilution, which no one else can. We can have an auto sampler. We haven't done the method validation of those calculations yet, because no one's really made us do that. So the measurement capability is there, the automation capability is there. I can't tell you that we've validated those calculations yet. So the automation is there, the validation support is not there yet, would be the answer to that.
Okay. The next question is asking you to talk a little bit more about sample dilution for SVI test less than 25 ml.
Sure, when we talk about sample dilution, that typically is when we're doing 787 testing. So with 788, in general, there shouldn't be any particles there at 10 to 25 microns. There shouldn't be particles there at 2 microns if you really have everything perfectly under control. With 787, where it's protein-based, and if we're dealing with high concentration proteins, then it is necessary sometimes to do dilution to stay below the concentration limit of the sensor. And typically what happens is it's a pre-dilution prior to walking up to and doing the measurement in the system. So what typically people will do is a 10-to-1 dilution, and we can actually put that pre-dilution factor into the software if we choose and get back to the actual concentration of the sample. That's probably a unique feature as well. So that's really a pre-dilution specifically for protein-based therapeutics, measuring using USP 787. And there the other trick is, what do you dilute with? Do you dilute with DI water or phosphate buffer or something else? And all I'm going to say is you'd better be filtering the heck out of your diluent, or that's going to end up causing you trouble as well. So the answer to that is really it's a pre-dilution. You have to be careful what you pre-dilute with. And we can enter that pre-dilution factor to get back to the actual concentration if that's where you're trying to get with that.
Okay, so I have another dilution question here, and forgive me if you've answered this. I'm such a novice at this. But the question is, does dilution impact aggregation by perhaps reducing the aggregation by dilution alone from a concentrated sample?
Well, there's somebody who knows something about proteins. So, I can look at a protein cross-eyed and it aggregates on me. Proteins are tricky samples, and anything you can do could cause aggregation, and you're right to worry about that. Remember, I would even mention I've done studies looking at the inner diameter of the tube or the needle I use to see if the shear stress of bringing it up through for the measurement causes trouble. So you're right to be worried about that. And to do dilution can cause aggregation if you're changing basically the physical chemistry, the surface chemistry in the sample. And this now will tie back to my previous response of what do you dilute with? And one should do studies of determining the optimum diluent to minimize that effect. So yes, you should be concerned, and the customer should be looking at diluent choice to minimize that. but should study it as well to make sure this isn't causing pass-fail concerns up at 10 and 25 microns. So it could, and yes, you should study it, and yes, you have to be careful to avoid it. But being very careful and given enough time, you can do this and generate excellent data. But don't expect to have that figured out in 2 hours. Give yourself at least a couple of days.
Okay, yeah, that was awesome. Thank you, Mark. I have one more question for you. How would you describe the level of compliance of software to 21 CFR Part 11 requirements? User privileges, security, audit trail, file security.
So there's a government document that says you have to do this, and then humans interact with that document. And that's where the complication comes from. So there's a document that says you have to do this. Now, each company that writes software has their own interpretation. And each customer could look at those requirements, have their interpretation. And so we have been very careful and have implemented following our opinions. And we've documented that very carefully in a document I briefly showed today. And it's not unusual before the installation or afterwards for us to share notes with customers and say, this is our interpretation. And it's almost a shock if someone somewhere in the conversation doesn't have a different opinion on something. So that's really where the conversation gets interesting. We certainly have reviewed the requirements. We've implemented the requirements following our best ideas, and we haven't run into any installation complications, but certainly if anyone is really deeply involved, they might say, well, we interpret it this way, and we'll say, we interpret it that way. In the end, everything works quite right, and all that ever happens is we have discussions over opinions.
I like the sound of that. So with that, that was our last question. Now, if anyone, had heavy fingers this morning and couldn't get their question out fast enough, or perhaps you have a lengthier question, I would like to invite you to email Mark directly. His email is
Right
right
there.
here on the screen. Ding, ding, ding.
And I love to
And
talk about particles. Feel free.
I can vouch for that. Mark, is there anything else you wanted to cover before I wrap it up?
Oh, we have all kinds of other documentation to support all this. We have application notes, we have white papers, I have data in articles. And really, if you find the stuff interesting, talk to us. Would love to talk about it.
Totally agree. So thank you everyone for joining. We appreciate you being here and bearing with us through our early technical difficulties today. We will be sending out a recording of today's event, and we will see you next time. Thanks again.
All right, thanks everybody for coming. Have a great day. Cheers.